Here in the second part of Chapter 19, we are going to take a look at critical issues that affect blood flow. Now, one of these things is going to be blood pressure, and we're going to look at that in more detail a little bit later on in Part 2. Let's start by defining blood flow. Blood flow is going to be a measurement of the volume of blood passing through a structure per unit time. We can measure it in mils per minute, liters per minute, etc. Blood pressure is the pressure that the blood exerts against the inside of the blood vessel wall. Blood pressure, again, the pressure that all of the molecules in the blood will exert against the inside of the blood vessel wall. And then resistance, I'm not sure that there's an easy way to define it without using the word resistance. Resistance is resistance to flow. In other words, anything that opposes blood flow is resistance found within the blood vessels. So next, let's take a look at a formula, a mathematical formula, that will help us understand the relationship between these things. Here is that formula. Flow is equal to delta P over TPR. Well, delta P is a change, that's what the symbol delta means in science, a change in pressure between point A and point B. So in our next slide, we are going to see evidence that there is, in fact, a change in pressure between the aorta as it comes out of the left ventricle and the vena cava as they return to the right atrium. The difference between those two is our pressure gradient. TPR stands for total peripheral resistance. So we are going to again take a look at the influences of resistance, and we'll look at that next. TPR stands for total peripheral resistance. In other words, we're looking at the sum of the resistance in all of the blood vessels throughout the periphery. And that's going to be directly proportionate to the length of a blood vessel. The longer the vessel, the more resistance. It's also directly proportionate to the viscosity of the blood. The thicker the blood, the more resistance. It is inversely proportionate to the radius to the fourth power. Now, we're going to look at that in more detail next. So, your textbook sort of interchangeably talks about the diameter of a blood vessel and the radius. And of course, we already know that the radius is half the diameter. It's the radius that we really talk about in vascular physiology, so let's take a look at that. Again, total peripheral resistance is inversely proportionate to the radius to the fourth power. And so I could write the formula this way. Resistance is equal to 1 over r to the fourth. R is the radius. So I'm going to prove it to you. If the radius is 2, I forgot to put the units, millimeters, then resistance will equal 1 over 2 to the fourth power, or 2 times 2 times 2 times 2, which equals 16. And so the resistance is 1 over 16. Now, let's look at if we double that radius and make it 4 millimeters. 4 millimeters. The resistance is equal to 1 over 4 to the fourth, or 4 times 4 times 4 times 4, which is 256. And so the larger the radius, the lesser the resistance. The larger the radius, the lower the resistance. The smaller the radius, the higher the resistance. And so, as promised, here is a slide that clearly demonstrates the fact that there is a pressure gradient. Here we have the aorta pressure, and here we have the vena cava pressure. And as we will see in more detail, not only later in this part, but also in lab, we see that in the aorta, arteries, arterioles, and just barely the beginning of the capillaries, we have what's called pulsatile flow. Pulsatile flow. And that means that there is a pulse. There is a difference between systolic and diastolic pressure. And you can see here 120 is our highest pressure. When the blood finally returns towards the heart and is in the vena cava, here you can see the pressure is nearly or at 0. So there is a difference in the pressures between point A and point B. And that is our pressure gradient, delta P. When we take a look at your textbook, it makes the point about pulsatile flow and the fact that we can palpate with our fingertips these specific locations where the pulse is palpable. And this illustration just demonstrates that. So this is a little blurry, but an image of an arteriolar wall. And again, it has the intima. There is an elastic membrane in the tunica media with smooth muscle. And then on the outside is the adventitia, or connective tissue tunica externa. So we need to know how flow is regulated. Well, primarily it is regulated by controlling the diameter or radius of the arterioles. To do that, there are a couple of different mechanisms. One is intrinsic controls, auto-regulation. This means it's local, okay? So in this case, the flow of blood is matched to the needs of very specific tissues. One of the mechanisms is a metabolic mechanism that involves paracrines. And if you'll remember, paracrines are signal molecules that are secreted by one cell and targets a cell very close by. So this is what we know. We know that if there is a decrease in oxygen in a particular tissue, if there is an increase in CO2 in a particular tissue, if there is an increase in acid, in other words, a decrease in pH, and if there are prostaglandins, which are local inflammatory paracrines, the result is that the act on the endothelial cells that line this arteriole, and that in turn releases a vasodilator. So is this a good thing? Well, let's think about it. You're exercising, say, one part of your body, and if that's the case, you are building CO2, Your oxygen in that tissue is going down. As you build CO2, that increases hydrogen concentration, which decreases pH. And all of these things are going to trigger vasodilation. The result of that will be increased blood flow. How? By decreasing resistance. What other factors influence arteriolar diameter? Well, histamines, which can either be released to target local tissue or can be released generally into the bloodstream to target a lot of tissues. Histamines cause vasodilation. If we apply heat to an affected area, that will cause vasodilation and increase blood flow. Or if you put an ice pack on an area, that decreases blood flow by causing vasoconstriction. And then the myogenic response we looked at in the heart. Basically, if a smooth muscle in the wall of the arteriole is overstretched, then it triggers vasoconstriction. So these are also three things that can influence arteriolar diameter intrinsically. Now, the extrinsic controls to adjust blood flow by adjusting the arteriolar diameter, I hope, is really familiar to you. Remember, we reviewed this at the beginning of the semester, that alpha-1 receptors are located on arterioles. They respond to norepinephrine, and they are excitatory. So that will trigger vasoconstriction. By the way, that also increases blood pressure, so we'll talk about that later in this part. Another thing to be aware of, though, is that there are override mechanisms for the sympathetic stimulation. For example, cerebral arterioles do not have alpha-1 receptors. This is ideal because we really don't want rapid changes in the diameter of cerebral arterioles. That would result in rapid changes in blood flow to the brain. So there are no alpha-1s in cerebral arterioles. And then secondly, the blood vessels that deliver blood to skeletal muscle and cardiac muscle are really strongly under the influence of those metabolic factors listed at the beginning of this slide. In addition, there are beta-2 receptors on these specific arterioles that deliver blood to the skeletal muscle and cardiac muscle. These beta-2s respond to epinephrine and are inhibitory. And so ultimately there is a nice balance between the alpha-1s and the beta-2s. We'll talk about that more in class. And as you will see in a later slide, there are hormones that influence arteriolar diameter. We talked very briefly about intrinsic versus extrinsic controls in Chapter 1 when we talked about homeostasis. If you'll recall, intrinsic controls are within the organ or the tissue. Primarily, paracrines are used, and this is often referred to as autoregulation or local control. Extrinsic controls from the outside of the tissue or organ, and those messages will be sent to the tissue or organ via nerves or hormones. Let's take a quick look at our intrinsic controls first. Let's take a look at some of these intrinsic controls, starting with paracrines. Paracrines are secreted by one cell and will target nearby cells. So what triggers these to be released? An increase in metabolic processes in cells. So when cells work harder, they consume more oxygen, they produce more CO2 as a waste product. Wherever CO2 is, we are going to see a change in pH, and so we'll see an increase in hydrogen ions in the form of carbonic and lactic acid, which lowers the pH. And then also local chemical messengers called prostaglandins. All of these target the endothelial cells of the arterioles, and they will trigger arteriolar constriction. Now, another mechanism that can change the arteriolar diameter is histamine. Histamine is a chemical released by inflamed tissues, and it triggers vasodilation. In addition, applying heat or cold can change things. If you put a heating pad on a limb, you will increase circulation, and if you put cold on, you decrease circulation. And then last, the myogenic response. Let me talk about that in another clip. The myogenic response of the smooth muscle found in the tunica media of arterioles is very similar to what we saw in cardiac muscle tissues. If you stretch these muscle cells, they are inclined to contract. And so the myogenic response is increased blood flow causes a stretch of the wall, triggering the cells to contract, causing vasoconstriction. Conversely, if there is low blood flow, then the muscles will relax, causing vasodilation. Our extrinsic controls, gosh, I really wish you had finished A&PII 2 before you came into A&PII 1. Now that's a joke. But later on in this semester, we will be talking about the autonomic nervous system. And there are two parts to that, sympathetic and parasympathetic. Now, their nicknames tell you more about it. The sympathetic division is called fight or flight. And so when you are frightened of something, your body responds with this mechanism. Otherwise, you are in parasympathetic mode, which is called rest and digest. Now, there are mechanisms that you will learn later in the semester that when you are frightened, when you go into sympathetic mode, vasoconstriction will occur. This increases some pressure and may increase flow. Another influencing factor are going to be hormones that we will take a look at in the first chapter of A&PII 2.